Welcome to the lab. I'm Alex Clooney. Today we will be discussing E. coli, in particular how to test for it. E. coli is very important to us as humans as we can be ill from it, with potential effects of diarrhea and vomiting, similar to that of food poisoning. Water quality was once first looked at by Dr. John Snow. Dr. John Snow lived in London in 1854, where a cholera outbreak came into the city. It was up to him to figure out where this was coming from, as many people felt it was simply bad air. However, they figured out that it was transferred through water over the course of time, and it was his job to track down the local pump. The local pump that was causing the issue was eventually quarantined, and he eventually saved many more lives. Unfortunately, Dr. Snow did not find this before there were 578 total casualties. These deaths were very concerning, and this is why we often look at many pathogens within water. Again, today the pathogen we'll be discussing is E. coli, and I will show you how we test for it in today's watershed quality systems. After incubating for 24 hours, we should be able to see the results. Before we go out into the field, we must have the proper supplies to go out to collect our sample. First, let's start with the test vial. The test vial will keep our collection sample after we have collected it. Before we can collect it, however, we use this pole to reach into our river or pond system, and we connect this beaker to the other end. That way we can collect a sample that is deeper in and less likely to disturb the overall sediment of the system. We also need to keep that on ice as we go, so we need an ice pack and a cooler. It's also important, again, to bring latex gloves. The main purpose is to keep you safe from taking any bacteria in the process. Hello again. We are back from the field now. As you can see, we've got our sample from the tech pond at the university, and we will be putting these samples back on ice before we analyze them as well. So now that we're back in the lab, you can see we have a wide variety of supplies laid out in front of us here. Let's start out with samples that we already collected. This is from the tech pond, and this is our tech pond 1 to 10 dilution. Now we have not actually mixed the 1 to 10 part all together yet, as we need to take 10 milliliters out of this one and transfer it into that one. We also need to, if you can see faintly on here, there is a line right about where my finger's running. That line means it's a 100 milliliter line. We will use this line to mark off after or before we put these reagents into mix. Let's go ahead and prep these samples now. Notice again that I am going to transfer 10 milliliters of our sample water into our other test vial. That was 5 mils. That was 10 mils. Notice I am closing the bottles as soon as possible in order to prevent contamination. However, as I discussed a minute ago, we need to clear this down to the 100 milliliter line, which I will do by lowering the water level with the auto pipetter. can see, the meniscus is now close to the water line. When these are matching, we can add the reagent. This is close enough, but you may not be able to see from this camera view as well as I can from the benchtop view here. We will now add reagent. Now as I shake this sample, I want to draw your attention to this machine right here. On the side you can see that it says Quantatray Sealer. That tray refers to those well sleeves that I had discussed earlier. Now what it is going to do is we will insert the Tecpon sleeves after the samples have been added, and those sleeves will be sealed shut. Now you might say, okay, well, why don't the sleeves seal shut on their own? They have an inside coating that allows for these individual wells to stay individual instead of water transferring from well to well. Therefore, if you want to imagine something like a hot iron going through and sealing it shut, that'd be very comparable. We'll be doing this in a couple minutes. The machine I'd now like to draw your attention to the machine that is behind the sealer. That machine is our incubator. The incubator is used to make sure that our samples are kept at a consistent temperature throughout the night. Bacteria like to grow between 25 degrees Celsius and about 45. Therefore we keep this at about 35, which is just below body temperature at about 96 Fahrenheit. Hello class, welcome back. We're going to read yesterday's results now. This has been incubating for 24 hours, and as you can see we've got yellow wells. These wells mean that I have total coliform growing. Total coliform, while it can be a nice indicator, is not an exceptional indicator when it comes to water quality. In large part because total coliform can be found on many surfaces. To determine our E. coli, we're going to be looking under black light radiation. Using the black light, we will determine the total number of E. coli wells lit up. This is the Tecpon sample from yesterday, and as you can see, there is not much there. Now, these 5 lit up wells up top, actually 6 if you want to include this one here, simply mean that there are 5 wells lit. That does not mean there are 5 E. coli bacteria there. Rather, we will have to go to a spreadsheet to compare later. Keep in mind, we have 6 large wells and 2 little wells lit up. Now, our 1 to 10 from the Tecpon yesterday will probably have less wells lit up. In large part, this dilution is not as strong as the other. We had a sterile buffer that we put in there in order to assure that the E. coli wells would not, E. coli cells would not lyse. As a result, we can see there's 1 and 2 little cells. These 2 little wells help us indicate that we indeed have less E. coli from sample 1 to sample 2, with sample 2 being the 1 to 10 dilution. We will look at the spreadsheet in order to determine the total amounts in our next lesson, but this is how you would check to determine E. coli and total coliform within a water system. Thank you, class. See you tomorrow.